The standard pH scale runs from 1 to 14, but human biology occupies a minute fraction of that range. Survival depends on maintaining a blood pH of 7.4, with a strict functional window stretching only from 7.37 to 7.43. Because pH is a logarithmic scale, it can mask the physical reality of the concentration we are measuring. This slide shows that a pH of 7.4 translates to exactly 40 nanomoles of hydrogen ions per liter of blood, or 0.00000004 moles. Human existence relies on preventing this specific, microscopic number of free protons from fluctuating. The biological paradox is that the process of staying alive actively threatens this baseline. Human metabolism acts as a continuous engine of acid production, as cells generate a constant stream of protons through energy production. This metabolic acid load is divided into two categories. Complete oxidation produces volatile acids, primarily carbon dioxide, which forms carbonic acid in the blood. Incomplete oxidation and protein breakdown produce fixed acids, such as lactic and phosphoric acid. To prevent systemic collapse, the body follows a strict requirement. The amount of protons synthesized by metabolism must precisely equal the amount of protons excreted. The body is under constant bombardment from its own metabolic products. It must absorb this massive acid load and clear it without ever letting the baseline proton concentration shift beyond the 40 nanomole limit. If the pH slips below 7.0 or rises above 7.7, the internal architecture of the body fails. Proteins require an exact pH to maintain their shape. Outside that window, enzymes lose structural integrity. This failure affects oxygen transport. Acidic conditions alter hydrogen bonds within hemoglobin, the Bohr Effect, causing it to release oxygen prematurely, impairing tissue delivery. Systemic acidosis also disrupts electrolytes. Excess protons force potassium into the plasma, leading to cardiac arrhythmias. Alkalosis is equally destructive, causing a drop in ionized calcium. This increases neuromuscular excitability, leading to muscle cramps, tetany, and seizures. A shift in pH is not just a chemical change, it is the physical trigger for cascading systemic organ failure. The body's primary, immediate defense against these shifts is the buffer solution. By definition, when an acid or base is added to a buffer, the pH remains stable, or changes only slightly. A buffer achieves this through an equilibrium between a weak acid, HA, which can donate protons, and its conjugate base, A-, which absorbs excess free protons. This chart maps the percentage of buffer components against the resulting pH, creating a sigmoid curve. Note the body's normal operating point on the flat central plateau. Here, the body absorbs large amounts of metabolic acid with almost zero change in pH. By maintaining its internal chemistry on this specific plateau, the body neutralizes the acid load and prevents fatal protein and electrolyte disturbances. To manage this chemistry, we use the dissociation constant, or KD. This equation defines the fixed ratio of dissociated ions to the intact acid in any buffer system. By rearranging this formula, we can isolate the proton concentration, making the variable that dictates pH the focus of the equation. To translate raw proton concentrations into the medical pH scale, we apply a negative logarithm to both sides of the rearranged formula. This results in the Henderson-Hasselbalch equation, the predictive model used to calculate how the concentrations of these chemical components determine blood pH. This formula proves that pH is mathematically predictable. You can maintain a constant proton concentration by managing the ratio of base to acid. Applying this to our primary defense, we replace the variables with our actual physiological buffer, bicarbonate and carbon dioxide. We then input the physiological constants. The pKa is 6.1, bicarbonate is 24 millimoles per liter, and normal CO2 partial pressure is 40 millimeters of mercury. These two variables represent organ function. The numerator, bicarbonate, is regulated by the kidneys, while the denominator, CO2, is regulated by the lungs. Calculating the ratio of 24 millimoles of renal bicarbonate against 40 millimeters of respiratory CO2 results in a pH of 7.4. The 40 nanomole limit of human survival is maintained by this continuous collaboration. By adjusting their outputs, the lungs and kidneys keep this single chemical ratio in equilibrium.